So wonderful to be here, and they've given me the task of waking everyone up or stopping you to go to sleep after lunch. I'm going to talk about some old work. Those who know it might fall asleep, but I promise to put in some new work here and there as well. So outline of what I'll be talking about so you can know when to sleep and when to wake up. I'll start with sort of the basic principles of the view I've espoused of evolvability, especially the pre-mutational shaping of cryptic sequences. Then newer work comes into, applying that to some data and finding evidence for it. Then the question of evolutionary capacitance, which comes into dual inheritance systems of how to tap into that pool. And finally, some very new work, very new work on how this is the mutational shaping on directional bias and what happens during how directional bias enters population genetic models to change the outcome of adaptation. So I'll begin with this earlier work on the theory of cryptic sequences and evolvability. So I'll start with an observation that seems to be pretty much universal, fundamental source law of how biology works. If you throw random mutations at something, you either break it or you tinker with it, and you don't often get anything in the middle. So the distribution of fitness effects of new mutations is bimodal. And those are some estimates from data. But now so this stuff is awful, and this stuff, by a process of elimination, can only be better from an evolvability perspective. And normally when we think about evolvability, the problem is, well, how do we know what variants are going to be adaptive, that's going to be highly context specific? But there are very, very, very few situations where being lethal or sterile, where major disruption of a developmental process is going to be adaptive. So while we are very poor at saying what is going to be adaptive, it's not nearly as difficult to say what is absolutely extremely unlikely to be adaptive. And then by a process of elimination, if you get rid of those, then you've increased the adaptive potential of whatever's left. And so when I started working on the field of evolvability, I looked at the literature, I saw a couple of definitions. One was the ability to generate heritable phenotypic variation. The other was the ability to generate adaptive heritable phenotypic variation. And I settled on a definition in the middle, which is to generate heritable phenotypic variation that isn't self-evidently awful, right? And that is this ratio of these two modes of the distribution of fitness effects of new variants. So the idea of a cryptic sequence is illustrated here using an example I've studied a lot, which is reading through a stop codon. So we have the regular sequence of a gene, and it has some effect that affects some trait, which might then feed into a pathway and so on. It doesn't have to be linear. But then if a particular thing goes wrong, if something happens at a certain rate rho, then in addition to this effect, you also get this effect, but this effect is small because it's multiplied by this small amount of expression, rho, but it also affects this trait. So that's a sort of a simple model of a cryptic sequence. And the key point is that this cryptic sequence can become a non-cryptic sequence through a process of mutation, called co-option here, where it gets co-opted. Now if this process happens, there's a new cryptic sequence, but meantime, instead of this only having a small effect, there are ways of triggering it to have a large effect instead. So when you have these co-option mutations, they aren't especially new. And the key insight is that when you have any kind of molecular or developmental error in the present, it tends to be congruent with mutations that might happen in the future. And I've heard a lot of talks at this meeting about that congruence, for example, between plastic responses and genetic variation. In the molecular realm, it's particularly obvious. You can see in the process of transcription, that recognizing the wrong nucleotide and making a one nucleotide mistake is exactly the same thing as happens when you replicate DNA, and you also make a one nucleotide mistake. You can see the mechanism of the congruence very clearly in those cases. But, if you have this congruence, then whatever's happening in the present tells you what future mutations might do. And then if you select on the consequences of those errors, then you can do this pre-screening where you have these two modes and you can get rid of the really bad variants prior to them appearing by mutation. And so that means that these cryptic variants that have been expressed a little bit already should have better properties than absolutely, truly new, non-pre-screened variants should have. And so you can illustrate that as a change in the distribution, and for this talk I'll just be calling that pre-mutational selection. So when you relax selection on these cryptic sequences, you get cryptic genetic variation. That's sort of the main idea of Waddington, which I've illustrated here, of 3 different individuals having 3 different cryptic sequences, and the light and dark blue colours here show what phenotype you would have if it were expressed, except it's repressed. And so what you get is only a marginally darker or marginally lighter phenotype with respect to what the environment is calling for. Here, if you have a slightly stronger effect, it might be slightly more visible. And then in some order or other, you have a co-option and an environmental change that collectively create a new match, and this cryptic sequence turns out to be really useful for adapting to the new environment. So this can be represented in a genotype space, and this is sort of the emphasis of Andreas Wagner on of these neutral spaces, that here are 3 different genotypes. This square is a population that is spread out among these neutral genotypes that are connected within this neutral network. And from each of these 3 different places, there's the possibility to mutate to a different place. But that isn't the only way of doing it. So far, that's just one gene. And we all know that most traits are controlled by more than one gene. So when you have a trait that's controlled by more than one gene, then within the same individual, you can have different cryptic effects. Even in a completely genetically homogeneous population, you can have heterogeneity among the cryptic sequences, which provides the possibility to adapt to different environments more easily, even in the absence of genetic variation. So we have these 2 views. You can think of one locus only, genetic diversity, accessing the ability to access one new phenotype with the neutral network perspective here, and then this other perspective, no genetic diversity but multiple loci, and that particular genotype can access all 3. And so we'll call them spread and neighbourhood richness. And what we found was that when there's compensatory evolution, when it's not just stabilizing selection to keep these things near 0, but the possibility that stronger negative or positive effects happen every once in a while, and rather than be reversed, they get compensated. When compensatory evolution takes place, you get a particularly rich filling of the potential of the cryptic variants. And because of this compensatory evolution, you get an overwhelming dominance of this as the cause of evolvability rather than the neutral network and the spread within it. So the neighbourhood, the richness of the mutational neighbourhood is more important than the spread within and the polymorphism within a neutral space. Okay, so that's the principles, the theory behind, with all the equations taken out, behind the pre-mutational selection. And next is some more recent unpublished work, although you're welcome to tweet everything, on whether it is in fact happening. And this is in our favourite study system of reading through stop codons. And the question we ask is, when you read through a stop codon, does a history of being read through mean that the sequence on the end, if it were translated, is going to be more benign, that you add that C-terminal amino acids on and you get a friendlier, happier protein with that isn't bothered by having those extra amino acids. And as a proxy for whether it's benign or deleterious, whether it's bothered by this, we use the intrinsic structural disorder, which basically, this central part, that's what ordered proteins look like. They have alpha helices, beta sheets, and they don't wobble very much. And intrinsic disorder is just a whole ensemble of states that move around a lot, interact with the solvent, and wobble. And the nice thing about this is there's a bunch of evidence and theory that suggests it will be benign, and very conveniently we are able to predict intrinsic structural disorder just from reading the amino acid sequence, which makes it suitable for high throughput comparative work. And you can see that it's benign by the fact that genes have high rates of intrinsic structural disorder. So here's the gene as a whole. Here's the slightly higher disorder that you tend to find specifically in the C-terminal tails. And here's the extensions. And actually, if I took junk DNA, it'll be down here somewhere. So genes, sometimes we think of proteins as being more ordered than you'd expect, but actually there's generally a lot less ordered than you'd expect from random sequences. Okay, so it is, being disordered is something that makes you pre-adapted to be a protein, and it especially makes you pre-adapted to be the C-terminal of a protein. So the question is, has there been selection on these to make them more disordered? They're not as ordered as the proteins are, but are they more ordered than they would be? And the way we ask that is to sort of exploit variation in the degree to which we expect selection to be working. Are the more strongly expressed ones, have they undergone more pre-mutational selection, and are they more disordered than the less strongly expressed ones? And to do that, we used ribosomal profiling data. So ribosomal profiling is a technique where you take the RNA, here are the ribosomes on it, you get rid of the free RNA that's in between, and then you get rid of the ribosomes, and then you're left with a bunch of little footprints where the ribosomes used to be, and you have a snapshot of what translation was at that moment in time. And what we find, in agreement with the prediction, is that when you find ribosomes, you get substantially higher intrinsic disorder than when there are no ribosomes. And the way ISD is scored, that might be contaminated by some differences in the main protein. So we did it again, but we grafted them all on a standardized set of proteins so that they're all on the scaffold and they're completely comparable. And even when we do that, we still see a substantial difference that's due to the sequences themselves. So sequences that have been under a greater, stronger history of pre-mutational selection do have different properties that predispose them, that pre-adapt them for co-option and for future genetic evolution. All right, part 3 is evolutionary capacitance. And so, if we've got this awesome pool of cryptic sequences, how does evolution exploit that? How does it get used? So evolution is the main term at this meeting seems to be inclusive inheritance. I'm going to talk about dual inheritance because just making it inclusive, doesn't really make a difference at this point. The equations of population genetics didn't know about DNA, don't care what the molecular structure or the physiological structure or whatever of the inheritance is. What's really interesting is that there are two inheritance systems that might have different properties. And so there's this genetic inheritance that can be cryptic or non-cryptic, and then there's this other inheritance system, the capacitor, that acts as an on-off switch, and it's the fact that things are happening on two levels that's the important part. And there are two advantages of doing this, and the one that's sort of discussed so far is that the quality of the variation, these variants in the cryptic sequences, has had this history of selection, and therefore it's much more likely to be ready to go. And then there's also the fact that the switch can be stress-induced, and so it can correlate the quantity of genetic variants with the extent of the need for rapid adaptation. So on both quality and quantity grounds, the evolution of a pre-adapted stock is something to exploit. And evolutionary capacitors are basically widgets that have adaptively evolved because of the history of selection of favouring evolvability to exploit such variation. And the particular one that I've studied the most, that's a beautiful system, is the yeast prion psi. And this is how it works. So imagine there's an RNA here, a coding region, a stop codon, a ribosome, and the protein in question is this pink one, and it's involved in recognizing stop codons. It's a eukaryotic release factor. And there's cryptic sequences are what there would be if you were to get rid of the stop codon and what effect that sequence would have. And that's in the psi negative system. And in the psi positive system, this release factor is bound up and sequestered in a heritable prion aggregate, which means that there's some amount of read-through, not just at one protein like there would be if you had a co-option mutation, but in all the genes in the yeast genome. And that leads, in a wild-type yeast, to a modest increase in the rate of stop codon read-through, something that the yeast can still tolerate, but that's certainly, increasing the degree of selection further on the phenotypes that are promoted by those sequences. And what's been observed is in stressful, there is direct evidence that this helps evolvability, like we see the yeast evolve faster, direct measurement. We have quantitative measures of switching. We know exactly, we can calculate the rate at which spontaneous or induced switching occurs. So one thing that's actually puzzled me at this meeting is, if you're interested in dual epigenetic inheritance, inclusive inheritance, and so on, there's amazing systems in microbes, like, cut and dried. I understand if you want to study humans and the cultural inheritance, great, but like other metazoan species, like why, right? Other than cultural evolution, there's cut and dry, there's nothing controversial. The really weird thing about capacitance is when it came out, there were two main model systems. The first one was HSP90, which turned out to be a really lousy system. And the second one was this psi system that's just beautiful. And the community embraced the HSP90 system and then ignored the psi system primarily, which says that Drosophila people care about these things and that yeast people don't primarily, and that says that the people who care about these things care more about studying metazoa than they care about evolutionary capacitance. So I think that's an interesting point when you're picking the model system for the thing you have in mind. Anyway, so yeast have dozens of these prions. They have an intricate non-DNA-based inheritance system, all of which involves, turning proteins on and off. So anyway, the way it works is you have these cryptic sequences beyond the stop codon. We have this pre-mutational selection, whether it's because of the basal levels of leakiness or whether it's because this prion turns on every once in a while. When the prion does appear, floodgates get opened, you get access to all of this different sequences at a higher rate than before. Some of them, of course, most of them are bad, and there's weeded out the worst of them. But meantime, some of them that are good, that were perhaps below the radar of selection here, are getting just above the radar. And then you get, strong enough selection such that the genetic assimilation, which in this case is the mutation in the stop codon, occurs. Genetic assimilation, this time not to an environmental perturbation but to an epigenetic perturbation, means that this adaptive phenotype has lost its dependence on that perturbation, on that presence of the prion, which means that the prion can disappear again and make the deleterious sequences go back to cryptic, and so there's no harm done. There's no damage of any kind done to the system. You know, analogy could be that, you know, you open up the cupboard, rummage through things that you put away there because you thought they might be useful at some point, find what you need, and close the cupboard door, right? Any disadvantage is strictly temporary. So it's a very neat system with exceptionally good data available for it. And so more broadly about these epigenetic or alternative systems, as I said, it's the same equations. You know, you've got the introduction, whether it's the price equation or a replicator equation or whatever, you've got an equation for some heritable change that enters into it, and the math doesn't care whether it's at the DNA level or not. But what is different, especially if you've got two systems with an interplay between them, is that they might have different rates and they might have different distributions of effects. And so then the question asks, well, whatever system you're interested in, be it prions or plasticity, with the environment being the way that is inherited, or whether your cultural evolution, whatever it is, you know, how are the rates and the distributions of effects distinct in the two cases that create the interplay? And one thing that tends to be the case for alternative systems is that they change at much higher rates. You know, genetic inheritance is extraordinarily stable, and the mutations are relatively rare. So that's sort of one difference that you'll find to be fairly general across systems. Another is that these alternative ones tend to be more environment dependent, right? That's an important part of what makes the math different when you put two of these systems together, and you don't just have more of the same, you have two things that really can interplay. And then something that, you know, really struck me, especially about the prion system, is that they're more symmetric. So when it comes to genetic mutations, we have all these beautiful, intricate, and extraordinarily fragile proteins. You know, a huge portion of mutations to them are premature stop codons, which destroy them, or deletions of some other kind, which also destroy them. We have intricate, information-rich product of adaptation that are just really easy to get rid of, right? That's how the distribution of effects is. But, you know, and if yeast were to go around jettisoning a protein as soon as it's not useful or maybe even harmful, you know, you end up losing an enormous number of proteins pretty fast. How do you stem that loss? So one possibility is that by having an even faster switching rate in which proteins are only temporarily lost, that removes the selection pressure to lose a protein that you otherwise might lose. If there's fluctuating selection on selection for presence-absence, you get an epigenetic switch to do presence-absence for it in order to prevent there being selection for permanent and irreversible loss. In particular, you know, when you have a microbial system, but without the rampant horizontal gene transfer of prokaryotic systems. So that's an important, you know, mathematical difference, you know, in the formal structures of the model. All right, so another thing that evolutionary capacitance helps with is crossing fitness valleys. So here's a fitness valley, you know, just say you have a peak and there's a new peak, or it would be a new peak if it was, you know. And then when it's cryptic, you know, the bad stuff's not as bad, and you can wander around the fitness landscape a little bit easier, and the valley's not as deep, and it might be easier to cross. And we and others have made this point. But the really striking thing that we did a bit more recently was, like, imagine we have, you know, the main thing that's wrong with fitness landscape metaphors is that we don't get our head around how high dimensional they are. You know, it's not just 3 dimensions and, you know, that in 2-dimensional a surface that you can define peaks in. You know, this huge number of dimensions. So if you've got n different places you can go, and then there's n choose j places you can go that are j steps away, right? And n choose j can be a very large number when n is huge and j is not small. And if each of those things has a little tiny chance of being an innovation, and the landscape otherwise gently slopes down, so you've got, you're at a fitness peak, you're at a local optimum, but somewhere j away is an innovation that would be even better if only you could get there, then what we find is the combinatorics of the numbers of peaks completely overwhelms the problem of the valley crossing, and valley crossing becomes not a big deal. Indeed, if you get that many dimensions, you can actually calculate that if the landscape's constructed this way and you adapt by revealing variation via a capacitor, you actually usually get a valley being crossed. Is more common than usually going to a local peak that's nearby. All right, so after that, so basically the central part of the capacitance story is that the capacitor turns on. The capacitor reveals lots and lots of variants, most of which are deleterious. So for the capacitor to be useful at a moment in time, it means that some one or several of those variants are sufficiently beneficial that it outweighs the cost of the bad ones. And that's sort of the condition on which the capacitor becomes useful. And when that happens, we have these systems like the prion that have evolved to take advantage of that and reveal when that takes place. But more recently, we also saw that even if you haven't evolved a widget like the psi capacitor, for utterly non-adaptive reasons, the same thing can take place anyway. If it's adaptive to do so, then you can get a regular genetic mutation as well that will increase the read-through rate, and then you will get genetic assimilation. And then depending on how hard it is to re-evolve a high fidelity the stop codon back, you can inch your way back to normality again, depending on just how asymmetric you made the mutations. So if it's not too difficult to regain fidelity, then you don't even need an adaptive widget. It happens spontaneously. And yes, a stopgap solution in all times that just hangs out until you find something better. Better to read through everything than wait around for the perfect mutation. And then this emergent capacitance is actually enough to take a system. I haven't shown some work we've done on when there's sort of two evolutionary contractors of a lot of these states, but it's enough to tip a system from something that doesn't have pre-screened cryptic variants into a system that does, because just while it's elevated, a whole bunch of purging takes place, and that makes it easier to get into a state where there's more purging in general. Okay, so finally, I wanted to talk about developmental bias. And this is very new, unpublished work. And the question is, so many people here are interested in developmental bias and measuring developmental biases. How is it that developmental bias synthesizes, if this is an extended evolutionary synthesis, it's not enough to say, here's this thing and here's this other thing and here's this other thing. It's necessary to synthesize them with each other and with a pre-existing theory. So how is it that developmental biases interact with the conventional population genetics theory? Where do they come in? So for this, just to clarify, I'm just talking about the adaptive mutations here. Developmental bias is about why one adaptive possibility takes place rather than a different adaptive possibility. It's no longer about the whole distribution like before. And I think the most important paper for this synthesis was this paper by Yampolsky and Stoltfitz, who are explaining how it could be that the process of introduction is an orienting factor that can explain what evolves. And to walk through what happens under a regime that the old name for it was strong selection, weak mutation. More recent authors, and I agree, think origin fixation is shorter and nicer. But anyway, the basic idea is imagine you're in the blue state and the green state or the orange state would both be adaptive. And one of them appears mutationally or otherwise at rate mu. And then once they appear, they have selection coefficients proportional to S. Okay? And the question is, which path does evolution take? When there is more than one path available, what happens? And what the result is it's saying that differences in mu are just as important as differences in S. And that's because the probability that something becomes fixed in the face of stochastic variation is proportional to how beneficial it is. And the probability that occurs at all is given by mu. And so you sort of have flux where things appear at rate mu and succeed with probability S. And so this product is the flux here, this product is the flux here. And you can see it's entirely symmetric that mu and S are equally important in seeing which one will take place. But the problem with this origin fixation dynamics is that it's only true when the total rate of beneficial mutation rate is small. So when two of these happen at the same time, that's not what happens. When two of them happens at the same time, the one with higher S wins. Because if they're both competing, there are processes, the one with higher S rises in frequency faster and outcompetes the other one. Unless they can be combined and synergistically become better than either one in isolation, S will rule unless this is true. When you are in this regime where this does work, the rate of adaptation is simply the rate at which things appear, which is the total rate of beneficial rates U, the number of individuals it can appear in, those people who, like diploids, can stick a 2 in if they like. And then the probability of fixation is S, and then the benefit to adaptation when it does happen is also S, and so that's the equation. And when U times N is greater than 1, then, as I said, there are competing selective sweeps, and there's been a huge developments in the last 12 or so years in population genetics of travelling wave theory. One of the simpler versions of that, early versions of that in asexuals, gives rise to this equation here. Selection is terribly important. There's an S squared term, but U is really not so terribly important in this equation. So what that means, remember, U is the developmental bias. U is which adaptive variants take place. If all you're interested in is developmental constraints, then obviously the things where U is 0 never happen. But if you're interested in more subtle developmental biases where either thing could happen, but one of them happens a lot more often than other for interesting developmental reasons, that's in different values of U, and that doesn't affect this equation very much. So here's a graph of the strength of selection. S is on one axis, U, the mutation rate or other introduction of variation, is on this axis. And here's the line, UN equals 1. Below this is in this origin fixation regime. Above this is in this travelling wave regime. I'll talk about this regime in a minute. But the question we ask is, okay, if I take a value of S, different values, a range of values of S, how do I vary U so that I always get the same rate of adaptation? All right, so how much do I need to change U to compensate for a change in S? So that's another way of asking how is selection important relative to developmental bias. And here's the equation for the origin fixation regime, drawn here in solid where it's actually valid, drawn here in dotted where it's not valid. And the fact that this has a very shallow slope is what indicates here that mutation-driven adaptation, the importance of developmental bias is possible, that you don't have to change U very much in order to compensate for what's going on in S. So shallow slope means, yes, you can have developmental bias as a cause. The steep slope, so then this is the equation for the travelling wave regime, and it does not have a shallow slope. It has a steep slope. Like I said, you get clonal interference, you get, and the thing with the larger S wins. But like the curve actually looks really weird if you plot it, and the equations say that you shouldn't plot it past this line. There's an approximation in the equation that says don't go there. But even before you go there, it's really interesting that it's starting to curve. So what we did is we did some simulations where we didn't have to worry. We have not solved this regime yet, but in the meantime, we looked to see what does happen. And while this isn't quite true here, the curve is real. And so what happens is you really do get mutation-driven adaptation coming back for extremely high levels of mutation. So I think I might be out of time, but what's happening very quickly for those who care is it's a travelling wave model. You've got selection on different fitness classes, beneficial mutations, feeding a nose, combining to get a travelling wave moving forward. This is how these models work. And you have the nose a certain distance from the mean. What happens in this regime is you get multiple stochastic classes at the same time. The ones that are further ahead have stronger selection on them, and they also have a lower threshold to escape stochasticity, which means that they can leapfrog up, and then you can get extremely sort of discontinuous sudden selection and huge saltationist jumps in fitness, even though you had tiny values of S and just really large values of U. So here is the summary of my points, since I am clearly out of time and people to thank.
Thank you.